Hello, everybody. What we're going to be working on today is finishing up our notes on genetics. We have basically two topics left. We're going to talk a little bit today about some human genetic diseases and a little bit about humans and how the genetics of humans and other complex animals are actually quite a bit more complex than simpler organisms. Like Mendel is studying with pea plants. We're also going to talk a little bit about sort of modern genetics or modern genetic technology. And then we'll be done with this section of our notes. So to start off, when we studied Mendel and worked on sort of Mendelian genetic problems, things were fairly simple. The traits that Mendel was investigating in his pea plants, they were simple dominant recessive traits. They were coded for by one single gene, and they were inherited in a predictable way. When we start looking at things a little more closely, however, we know that things are more complicated than that. For example, in human genetics, we think about human traits and characteristics, eye color, hair color, things like that, height, personality traits. Things are more complex. It's not like there's just one single gene that has two alleles for hair color, and it's dominant recessive, and you can easily predict the results of two parents having offspring. Most human traits are much more complex because they are coded for by multiple genes. Okay, so things like your height or your eye color, they're not simply controlled by one gene with two alleles, one dominant, one recessive. Many times there are multiple genes which result in certain human traits and characteristics. And so it's the interplay of those genes along with the effects of the environment that influence those traits. And so I can't really give you very many simple examples of a human trait that you can do a Punnett square with your parents and figure out what the likelihood of their offspring are. It's more complicated than that. We sort of simplify things when we talk about Mendelian genetics to help us understand sort of patterns of inheritance, but typically things are more complex than that. So one of the things that we'll talk about are some human diseases that are caused by defective genes. Okay? And so there's a host of these, and we could go on with a list of human genetic disorders. We'll talk about just a few of them here, and you could research others if you're interested. But we can now sort of detect these genetic disorders using certain techniques of modern genetics where we can actually test and see if a person has certain genes. And there's other ways to do this. Sometimes screening, looking at the symptoms a person has, taking samples of fetal cells with an amniocentesis, creating karyotypes, genetic testing. These are all ways of trying to determine whether or not a person has a certain trait. So one of the genetic disorders we'll just mention it's abbreviated PKU, phenylketonuria. And this is a disease that's actually caused by a recessive allele. So it is a recessive allele. If a person inherits two recessive alleles, then they will have this disease, PKU. And what happens is that because there is a person with PKU has a change in the usual gene that allows their body to produce an enzyme that breaks down a specific amino acid. One amino acid is called phenylalanine. It's one of the 20 amino acids. And in a healthy individual, they have enzymes that can break down that amino acid. However, a person with PKU, there is an abnormality in the gene that produces that enzyme, and therefore the enzyme doesn't work properly. Okay, so what happens is this amino acid phenylalanine can build up in their body. It can impair the neural development of an infant as they're developing. It can be detected using genetic testing, but also can be detected traditionally by analyzing a urine of a newborn. And if it turns out that a person does have PKU, they have to eat a very strict diet in which they don't eat this amino acid phenylalanine because they have no way of breaking it down. And so, again, this is a genetic disease that basically we can look at the pattern of inheritance. If the parents are both carriers, as we see in this pedigree, then they will have a 25% chance of having a child that has it, because there's a 25% chance of the offspring being homozygous recessive. Another genetic disorder is sickle cell disease. Sickle cell disease is again caused by a recessive gene, and it is a disease that's much more common in people of African descent. And when a person has this disease, sickle cell disease, and if they inherit it, two recessive genes, then it results in the hemoglobin molecule that's in their red blood cells being incorrectly formed. Okay? And those red blood cells don't carry the proper amount of oxygen. They become misshapen. Okay, you can see on this picture, this is a sickle cell blood cell. And as they flow through capillaries, they can block the flow of blood, and lead to, extreme pain and organ malfunction. And so, again, because this is a recessive disease, a person could be a carrier. They could be having one of the normal genes and one of the sickle cell genes, and they wouldn't know that. And it's an interesting story about sickle cell disease because being a carrier, having one of these genes, is actually an advantage to people living in areas where malaria is endemic. Okay, a person that is a carrier for sickle cell disease actually has significant resistance to malaria. Okay. And that's one of the reasons it's likely more common in people of African descent. There's not a cure for sickle cell disease. People often get blood transfusions and hopes of regaining function of those normal red blood cells. But there are newer treatments that are sort of being studied currently of using modern genetic manipulation to change that disease recessive gene and to try to make it into a functional gene. Another example is called Tay-Sachs disease. This is again a recessive allele that leads to again a problem in the production of an enzyme that helps the buildup of fats in the brain. And Tay-Sachs disease leads to the deterioration of neural tissue. Okay, and it is more common in people of Central European and Jewish descent. And this can be tested for. You can see here we have the genetic sequence of the normal allele for this enzyme and the Tay-Sachs allele. And so what you're going to see here, okay, is typically the gene is TCCTAT. Well, what's happened is, okay, there is an alteration here, okay, in which a gene has been deleted. A base has been deleted. So if we delete this C, okay, from the sequence, everything gets shifted over one. Okay, we have TAT is shifted over, and we have then a misshapen enzyme that doesn't function properly. This can be detected again through blood testing and through amniocentesis. Often people that have a family history of Tay-Sachs disease might be tested before they decide to have children to see if the parents are carriers. If the parents are carriers, then again, there's a 25% chance that their offspring will also inherit the actual disease. So we're going to move on a little bit to talk about sort of modern genetic technology. And we're not going to be able to get into the details of lots of this. We'll sort of give an overview, a background of genetic engineering and modern tools. There's actually recently, if you ever have heard of the documentary filmmaker Ken Burns, he does documentaries all the time and are really well acclaimed, and he just did one on sort of genetics, and it's called The Gene. It was a book that's really interesting, and his documentary talks a lot about modern genetics and the history of genetics. If you have an interest in this, you could check it out. It's on PBS. You could stream it. It's called The Gene, an intimate portrait, I believe. So genetic engineering is a type of technology that has been developed over the last 30 years or so, maybe a little longer than that, in which scientists can manipulate the DNA of organisms to a degree that is pretty amazing. And so this topic, genetic engineering, is kind of just an overall term that relates to a whole bunch of different things, and we'll talk about some of those. So one way of thinking about genetic engineering, it's actually altering the genetic code of an organism. Okay, and so the way this is done is by using special enzymes. Okay, they're called restriction enzymes. Restriction enzymes are special enzymes that cut DNA at a specific sequence of bases. Okay, and scientists have a whole suite of these restriction enzymes that they can use to cut DNA at different places. And this is useful. You could think of them as sort of molecular scissors. They actually go in to the DNA molecule, snip a section out. Okay, and so in genetic engineering, scientists identify a gene that they're interested in. They could use specific restriction enzymes to cut that gene out of an organism. And then they can take that gene and actually put it into another organism. Okay. So an example that's sort of the go-to example of how this has been used in medicine is in the production of insulin. So insulin, if a person is a diabetic and they need to administer themselves with insulin regularly, before the advent of genetic engineering, they would inject insulin basically that was harvested from pigs, from the pancreas of pigs that had been slaughtered for meat. And it was difficult to obtain enough of this insulin. There were sometimes problems with allergies and didn't quite work as well as human insulin did. And so it was a very difficult process to obtain that insulin. But when scientists figured out how to use this process of genetic engineering or gene splicing, they used it. And one of the first applications of this was to produce insulin. And what they did is they took from human cells, they identified the gene that tells our body how to make insulin, that has the recipe to make that insulin. They used a restriction enzyme to cut that gene out of the human DNA, and then they pasted that DNA, that gene for insulin, into a bacteria. And so once they've done that, the bacteria now actually have a section of human DNA in their genes. And as those bacteria grow, they actually use that gene and they start producing human insulin. Scientists can grow these bacteria in huge vats, basically, and these bacteria can produce an almost unlimited amount of insulin. And that's where our insulin that people use today, it comes from these genetically engineered bacteria that are making that human insulin because they've been genetically engineered. It's used to make other human proteins as well, like human growth hormone and so forth. And this has been done many, many times for various uses, and we'll talk about some examples of that. But again, here you see the DNA molecule with a sequence of bases. A restriction enzyme can cut it open, and then in that gap, a new gene can be placed. Scientists now can read the genetic sequence of an organism using automated equipment. For example, when we're talking about the coronavirus, scientists have been able to study the DNA of the coronavirus to understand how it's evolved, how it changes, how it works. Scientists can actually create DNA strands synthetically by attaching purposefully different bases in a specific order. So this diagram basically shows a pictorial way of how humans produce proteins using these genetically altered bacteria. This is another example. This is using them to produce growth hormone. It's the same exact process that's used to produce insulin, but you have a human cell. You find the gene that you're interested in. In this case, they're looking for the human growth hormone gene. They use a restriction enzyme to cut out that gene from the human DNA. At the same time, they take the bacterial DNA, this circular DNA called a plasmid, they cut it open. They insert the human growth hormone gene into the bacterial plasmid, put it back into a bacteria cell, but now, as you see, in the DNA of that bacteria, they actually have the human gene. And as they grow, they will begin producing those human proteins. So that bacteria would be an example of something we call a transgenic organism. It's an organism that has been produced with DNA from another organism. Okay. If you ever have heard about GMO foods, for example, that stands for genetically modified organisms. Some people are hesitant to use genetically modified organisms as products, and there are questions that sometimes people have about safety or allergies or ethics that are related to genetically engineering organisms. But here's just some more examples of how humans have used genetic engineering and gene splicing. I just talked about the first one, transgenic bacteria, where human genes are inserted into the bacteria to allow those bacteria to then produce human proteins. Humans have produced transgenic animals. For example, if scientists have a gene they want to study in humans, okay, they can insert that gene into a mouse model or some other organism, grow those animals, and then use them for experimentation to test different treatments and things like that. We have livestock, okay, that have been genetically engineered to produce excess growth hormone in their tissue. And that leads to them growing to a larger than normal size, increasing their muscle mass, which allows them to produce more meat. There are animals in which gene splicing has been used to allow them to produce medicines in their milk. Many plants and crops have been genetically engineered. Some crops have been genetically engineered to have inside of them a gene that produces insecticides, so that these crops are less prone to be eaten by insects. famous example of a transgenic plant is called the Roundup Ready soybeans or other cereal crops, in that scientists inserted into these crops a gene that makes them resistant to an herbicide called Roundup. You may have it in your shed or something. Use it to kill grass on the sidewalk, things like that. So because they've engineered these plants to be resistant to Roundup, what they do is they can grow a whole field, for example, of soybeans, and then spray the entire field with Roundup. Now, because these soybean crops have that resistant gene, they're not going to be damaged by the Roundup herbicide that's sprayed on them, but all of the weeds and so forth will be. And so this is a way of sort of mass treating fields with herbicides without harming the crops. Obviously, one of the trade-offs is that means that in agriculture we're using vastly more herbicide to grow those plants, and that has its own consequences in terms of the ecosystem, and we'll talk more about that in our next unit. Rice, a type of rice called golden rice, has been genetically engineered, and this golden rice has been genetically engineered to produce greater amounts of vitamin A precursor molecules, and so it's more nutritious, and it's helped to reduce blindness in populations that subsist basically on small amounts of rice. Typical rice doesn't have enough vitamin A to provide as much as a person needs, and so a lack of vitamin A can lead to damage to the eyesight. Growing instead this golden rice produces more vitamin A, helping those people that might be deficient. So these are some examples of how scientists are using genetic engineering. And now you may have heard in recent times about a new technology called CRISPR. CRISPR-Cas9 is basically a set of tools, basically chemicals, in which scientists can target a specific gene in a living cell and replace that gene with a corrected copy. So it's an idea about how we might be able to use genetic engineering to help cure disease in individuals. For example, this diagram shows a woman that has sickle cell disease, it's caused by one single mutation in the DNA. So scientists can use CRISPR-Cas9, this set of chemicals, to target that gene, that damage. Okay, they can add to it sort of a corrected version of the gene. They can inject that into a person, and then that CRISPR-Cas9 enzymes will insert the healthy corrected version of that DNA in where the incorrect version was. And the idea is that it can help to actually cure a disease like sickle cell disease. Now, these are early stages for this sort of treatment, and there are some diseases where it's being studied and being studied successfully. There are also examples of gene editing within a person that have had negative effects as well. And there's a famous example of a young man that died after an experimental treatment with a genetically modified gene that helps cure a disease. That was before CRISPR-Cas9. And for a while there was sort of a moratorium on that kind of research, and now it's starting to pick up because we have these new tools that possibly can make it safer. Cloning is a technology that was first done in the late 1990s. It was the first cloned mammal. Basically, cloning is producing an identical copy of another organism. Okay, and so the first cloned mammal was a sheep called Dolly. And in cloning, what happens is basically a nucleus is withdrawn from a cell, of the organism that you want to be cloned. So let's say I wanted to clone myself. That would be great to have two of me. One of me could be recording this video, the other could be playing a video game or something. But anyway, if I wanted to clone, for example, myself, I would have to get a nucleus from one of my cells. I could just use a cheek cell and take that nucleus out of the cell. Then I would need an egg cell, okay, from a female. And from that egg cell, I'd actually remove the nucleus that it had, get rid of it. And instead, I'm going to replace that with my nucleus from one of my body cells. Place my body cell nucleus inside of this egg cell. I'd give it a little electrical shock, cause everything to sort of adhere together. And then that cell would begin dividing. It's going to divide into an embryo. Now that embryo is a genetic copy of myself. It has all of the genes, all of the chromosomes in my body cells would be in this embryo. If you then take that embryo, implant it into the uterus of a surrogate mother, it'll grow, be nourished by the placenta, eventually be born, and that baby that would be born would be my clone. It'd have my exact genetic information. And so that's how cloning is done. Now, cloning can also be done not to produce an entire organism, but to clone tissue. For example, if a person is studying cardiac tissue and different cardiac diseases, they can take a tissue sample from a person that has a specific cardiac illness, genetic predisposition to something, and they can actually clone just that tissue, not to grow it into another organ, but clone that cardiac tissue, and then they can experiment with it to try to find treatments for those conditions. One tool, if we were in school, that I would show you and we would do is something called gel electrophoresis. Gel electrophoresis is a way of separating the DNA into groups according to the size of the DNA. And this is used, if you ever heard of sort of DNA fingerprinting or DNA profiling, if you watch any crime shows, things like that. Gel electrophoresis is used to produce some of those DNA profiles. And what you do basically is you take a sample of DNA, you add to it a restriction enzyme, which, if you remember, cuts the DNA up in certain sequences. And then what you do is you take that mixture of a bunch of chopped up DNA and you put it in a gel. It's like Jell-O, basically. It's this clearish gray gelatin, and you pour it into a tray. Okay, the gel is in here, and then you put it in this container that has a solution, electrolyte solution, and then you run electricity through it. And the DNA strands start to migrate through that gel. Okay, DNA is negatively charged. So it starts moving towards the positive electrode. And small molecules can sort of wind their way through the gel very quickly, and they get far. Large molecules of DNA don't get as far. And so after you run it for a period of time and you stop it, you stain the gel, and the DNA forms these bands. Okay, and the bands are made up of DNA that has been cut up into different sizes, and it forms a pattern. And so if you compare two different DNA samples that were treated with the same restriction enzyme, they should form an identical pattern when gel electrophoresis is done. And you're going to be doing an activity next week in which you'll do an online simulation of electrophoresis. Here again is a description of how it works, where the DNA samples combine with restriction enzymes to cut it into small pieces. Those pieces are placed in these wells. The different samples are placed in the wells. We add the electricity. The DNA starts migrating. We stop it, stain it, and then we have this pattern. The smaller fragments get farther towards the positive than the others. And you could use that for things like paternity testing, for example. Okay, it can be created using samples of tissue from blood or cells, even saliva, hair, semen. So that's why you see it's used sometimes in those crime shows as a forensic method of figuring out who was at the scene of a crime and so forth. Here's an example of paternity testing. So if this mother had a child but was unsure of the father, so like Mamma Mia here, was unsure of the father, this DNA profiling using electrophoresis could be done to help identify who the father was. So if you said there were 3 potential fathers of this child, what you do is you would take samples of DNA from the mother, the child, and all the possible fathers, and you'd run them through this process of electrophoresis. And what you would see is the pattern of bands that shows up in the child. Because every one of these bands that shows up in the child must be found in either the mother or the father. So when we look through the child's pattern of banding, well, this band probably came from the mother. This band probably came from the mother. This one did not, because the mother doesn't have a band right there. Same thing for these 2 down here. So these that don't match the mother must have come from the father. So if you then compare the potential fathers, you would look for, okay, well, could this have come from the father? Well, not that one. Well, it could have come from this one, could have come from this one. Same thing here. This one could have come from this father, not this one, could have come from this one. Same thing with this third one. No, yes, yes. This father, father number 3, has the pattern that matches up with all of those missing bands in the child. We would say most likely dad number 3 is the father of this child. And the same concept can work for, a crime scene investigation or something like that. If you find a blood sample at the scene of a crime, you can do a DNA profile of that blood sample, compare it to the profiles of several suspects, and see which is a match. All right. So that's the end of our notes here on modern genetics. And this is an interesting topic. When you go to high school, you might take a class. There's a class in biotechnology. It's really interesting about lots of these topics. Genetic engineering, gene splicing, CRISPR-Cas9, tissue culturing. These are all really important areas of study. Especially going forward, though, you guys, as you get into college and beyond college, if you go into medical fields or the fields of science, this is going to be really, really important ideas that are going to become more and more important in health and medicine and so forth. So if you find these ideas interesting, then it might be worthwhile looking into them a little more and thinking about taking some of those courses in high school and maybe pursuing those sorts of things as you move on to college. Okay, so I hope you guys have a good rest of your day. Have a great weekend. We've got some activities coming up next week. No more notes. And we'll see you later.